Annual Center for Drug Discovery and Development Summit. I appreciate all the everybody who is now Zooming with us. I think we have a total around 75-ish, and so that's a great turnout. Please, everybody, be careful with your mute on your Zoom today. We began our series of programs in 2018 and accumulated a great group of speakers over the past several years. We value your feedback. You're welcome to reach out to either co-director of the Arizona Center for Drug Discovery, Celina, Zerbinatti, or WeWound, with your thoughts on future programs. Program attendees, you're invited to submit questions during each presentation using the question and answer at the bottom of your screen via the question and answer feature. Good morning. We will attempt to get all of your questions. All unanswered questions will be collected and responses will be recorded and shared with attendees after the summit. So if there are no questions, we will proceed to the next speaker, our first speaker. I would like to welcome our morning moderator and co-director, Dr. Celina Zerbinatti.
Good morning, everyone. It is my great pleasure to introduce our opening speaker, Dr. Joseph Mikhael. Dr. Mikhael is a professor in the Applied Cancer Research and Drug Discovery Division at the Translational Genomics Research Institute at TGen, and he's also the Chief Medical Officer of the International Myeloma Foundation. Dr. Mikhail went to medical school in Ottawa, Canada, and obtained his residency in hematology at the Princess Margaret Hospital in Toronto. He followed his residency with a multiple myeloma fellowship, along with a master's degree in education from the University of Toronto. His clinical expertise is in plasma cell disorders, and he is a principal investigator of numerous clinical trials, primarily in multiple myeloma. His other clinical research interests include pharmacoeconomics, communication skills, and media relations. He's published over 150 peer-reviewed articles in these fields, and is an active member of the International Myeloma Working Group, serves on the editorial board of the Journal of Clinical Oncology, and lectures internationally on a regular basis. Dr. Mikhail is a consultant hematologist and director of myeloma research at the Honor Health Research Institute, where he conducts phase 1 clinical trials. He also serves as counselor on the executive of the American Society of Hematology, and he is leading an important initiative at the International Myeloma Foundation, the African American Initiative, which seeks to improve the care delivered to African Americans with myeloma. It is a disease that actually affects the African Americans twice as much as Caucasians. He also spends about 20% of his time in third world countries, seeking ways to enhance access to myeloma therapies for underprivileged patients. I want to thank you, Dr. Mikhail, for his incredible dedication to myeloma patients around the world, and for taking some time from his important clinical and busy work schedule to be here with us today. And he will be speaking on drug development and multiple myeloma from the clinic to the planet. Welcome, Joe.
Thank you so much, Celina. That was much too long and kind an introduction. I usually just say, my name is Joe, I'm here to give the talk, but I appreciate your kindness to me. And I trust that this next 45 minutes or so together will be helpful to all of those who are listening to provide some information and give a context of real drug development in the real world, but also to try and help us understand that we can look beyond our borders to many places around the planet where there is even a greater need. And this is a perspective that I try to bring to all that I do to help us appreciate what we have. We are so blessed in so many ways with so many things. And so hopefully today will be educational and informative, but may also be slightly motivational in the way you think about how we can help other underprivileged individuals and indeed other underprivileged countries. So We've tried to give it a bit of a catchy title, drug development of myeloma from the clinic to the planet. It sounds a little ominous. I'm sitting here in my clinic at the Honor Health Research Institute between patients, but I hope to take you on a little bit of a journey, as it were, around the planet as well, if that's at all possible. So my conflict of interest, I have served as a consultant for the companies listed here. And then what are our objectives? So you may know nothing about multiple myeloma. You might hear the word myeloma and think melanoma, which is skin cancer. And I'm sure some of you thought that, but I'll demystify that a little bit. So I thought I would just give you the quick primer in myeloma, try and give you some of the science behind it for those science geeks that we are. And also in that, talk a little bit about the development of novel agents over these last 20 years, a little bit on imaging and diagnostic testing. But I want to use myeloma as an example of a disease that we have doubled, if not tripled, the average survival of patients over the last 10 to 15 years. It has really been, although, as I'll show you, a relatively rare cancer, one that is, I would argue, a template for drug development. I mean, it's remarkable. And I have the privilege of being involved with many of these drugs previously in Canada, as was mentioned, but also at Mayo Clinic here in Arizona for 10 years before joining the International Myeloma Foundation. And then what I'd like to do is take that, sort of looking back a little bit in the history of what we've been able to do in changing really the face of this disease in many respects, still remains incurable and is still a tragic disease where we lose many patients a year, but nonetheless show you some of that progress. And then try and have you see that what we do here can help people elsewhere. And I'm very committed to spending a lot of my time in the third world, albeit right now mostly virtual. I've been to about 4 virtual third world countries this week. I guess in some ways I can get to more countries when I travel there virtually, but it's never the same as being in person, of course, just like this meeting. I really hope we'll be able to do it in person. But nonetheless, we'll go internationally and see a little bit of how this may work. And I'm just going to give you one model, what we call the network model, that may help facilitate drug development in these places. So for those of you who know a little bit about myeloma, this may be of value to you. For those who don't, I want to introduce you to it. There are going to be themes that pervade this talk. I was saying to the team earlier, I don't want to sound like Charlie Brown's teacher today, wah, 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 just going on and on and on. I want to make this as dynamic as a Zoom call can be. And so there are themes that are going to emerge today. The first theme I won't have a lot of time to talk about, but although this is a rare disease, as I'm going to share with you, it's less than 2% of all cancers. I can make an argument that maybe most common cancer in the world, and I'm not here to creep anybody out or scare you, but the pre-myeloma condition, there's this condition that I'll show you called MGUS, or monoclonal gammopathy of undetermined significance. I know it's a mouthful. It just means you make an abnormal protein in your blood. 5% of adults over the age of 30 or 40 do this. So it's incredibly common, but only a small fraction of them will go on to truly develop myeloma. It's analogous to, maybe having a couple of abnormal skin cells that may make a mole that someone says, oh, that could become a skin cancer. And sometimes we cut them out and sometimes we don't. It's the same kind of concept. It's a precancerous condition, but it's extremely common. And that is unique to myeloma so that we can learn from the precondition to understand the true condition later. Myeloma, I'm going to show to you, is a prototype of immunotherapy. If there's anything you hear at any cancer conference on the planet right now, immunotherapy, where we can leverage the immune system. And I got to talk to you a little bit about that and why I think it's so important in light of that first theme. I mentioned already in my introduction that the survival improvement of myeloma rivals just about any cancer in the last decade. And that's exciting because we want a bit of a success story. I want to be able to show you that we've made a difference. And this is a result of a huge host of new therapies coming at the disease at different angles, looking at different mechanisms. For those of you, for example, who followed the story of HIV, we've not totally conquered HIV, but we've made huge strides, not because we found one magic drug, but because we've leveraged multiple mechanisms of action together to overcome it. I'll talk a little bit, not as much about the diagnostic approaches, because this is a disease where typically we have to put a needle in a patient's bone marrow in their hip, typically, and draw out blood. But there are different ways of both diagnosing this disease through blood testing and imaging. And then I can't help but mention, as Celina noted that I do a lot of work within the African-American community. I myself am African-American in origin, and I'm very sensitive to this, that this is a disease that's twice as common in this population, and yet survival outcomes are about half of that within the Caucasian population. And so this is a pressing issue, and the events of the last few years have perhaps sensitized us to the need of understanding disparities better. And then I want to end on a positive note, because really this is promising. I've been doing myeloma for 20 years. Pretty well only myeloma for 20 years. And I have never seen a more exciting time, honestly, than this year, 2021, in terms of the opportunities. And I'll share a little bit about that. So just as a quick primer, so myeloma is a cancer of the plasma cells. Now, you probably don't have a picture of your spouse's plasma cells or your partner's plasma cells next to your bed, but what are plasma cells? Plasma cells got a lot of press this last year, because these are the cells, of course, that make our antibodies or immunoglobulins, of which, as you know, there are several types: immunoglobulin G, A, M, D, E. And so these normally function, they live in the bone marrow, they're normally less than 5% of your marrow. And when you get a vaccination or some kind of stimulation to the immune system, the plasma cells respond and make that antibody and store that antibody so that God forbid you get infected, those antibodies can be initiated and take down the infection. Well, what happens in myeloma is those same those same plasma cells that are meant to fight infections and make good antibodies, as it were, now make abnormal antibodies or monoclonal antibodies. Instead of being polyclonal, different shapes and sizes, now they're monoclonal. And this becomes obviously a huge problem. As I mentioned, it's only about 2% of all cancers. The average age of diagnosis is about 69, 70, but it's 4 or 5 years younger, interestingly, in both the Black and the Hispanic population. And so we do see a disparity even in diagnosis. I mentioned earlier that survival is continuing to improve quite dramatically. And even, as you may know, SEER, this only goes back to 2016, SEER databases are a little bit, behind because they collect information so thoroughly. But we have really seen dramatic changes where most patients now are going to live over a decade with this disease. When I started myeloma 20 years ago, most of our patients didn't live 1, maybe 2 years. And at that, they were an awful 1 or 2 years with broken bones and kidney failure and infections. We've made a long way to improving not only the quantity of life, as I'll share with you, but the quality of life. And I say it all the time, I don't treat myeloma, I treat people. And so we have to look at every patient indeed as a person. Now, this may look a bit of a busy slide, as some say. I usually say, don't make busy slides. They're actually called lazy slides. But I do this slide purposely. So I want to, for the immune interest group parties here, I'm going to make this very simple. I want you to just notice that there are 3 columns on the right here that are under the title of cancer immunoediting. This is a model of how, unfortunately, cancer can develop within our bodies. And it's based on 3 functions that all start with the letter E: elimination, equilibrium, and escape. And you'll see the relevance to myeloma and to the whole talk in just a minute. So elimination, what is that? That means that your immune system, while you're listening to me today, sitting in your pajamas, drinking your coffee, or doing whatever you're doing today, your immune system is vigilantly trying to ensure that none of your cells become cancerous. Trillions of cells in your body are replicating as we speak. And so there's the potential for malignancy is extremely high. Just think of the number of transactions that are occurring. You make a couple of tiny errors and you can have a skin cancer, you can have a prostate cancer, you can have a breast cancer. And so your body is constantly trying to eliminate that risk. Kind of like the teacher that is noticing that one of the students is looking out the window in the middle of lecture and says, hey there, Billy, look over here. I don't want you to miss out on this. And you have that component to your immune system. It's a beautiful thing that has that ability to reduce the tendency towards malignancy. That is a centerpiece of immunotherapy. Part 2 is what's called equilibrium. This is when the immune system can't entirely eliminate, but can at least keep in equilibrium small amounts of cancer. So here a few cells kind of go a little bit rogue and they create a little bit of cancer, but the immune system is able to keep them in check and say, okay, look, you're allowed a little bit, but it's not going to be a big problem. I kind of joke and say it's a little bit like having a bit of dessert, right? We all need a little naughty in our life. If you don't have a little bit of dessert once in a while, you go crazy at the buffet, right? So this, interestingly, and I can show you even in a model in our lab with mice, I can put a tiny tumor in a mouse, and that little bit of tumor, paradoxically, can actually protect the mouse from getting a more aggressive tumor. It's a very weird phenomenon, but it's there, and we call it equilibrium. And I mention that because this concept I introduced earlier of MGUS, or monoclonal gammopathy of undetermined significance, is really an equilibrium that almost 5% of all adults are currently doing. You've got this little schmutz of abnormal cells and abnormal protein in one's blood, but it's not hurting anybody. And we're not going to do anything about it. Imagine a similar, let's say, men with prostate cancer. If I go biopsy the prostate of every man in the country over the age of 70, I'm going to find a lot of tiny bits of prostate cancer. We're not going to do anything about it. We're not going to go take it out with surgery or give radiation or give chemo, because it's so small, it's never going to hurt anybody. It's like finding a little bit of dust in the corner of your room. It doesn't mean you run and get the vacuum cleaner. It's just a small amount. It's not going to hurt anybody. But if it grows, obviously that's a problem. So this concept of equilibrium is important. And then lastly is what we call escape. And that's when the immune system can't hold on anymore. And it's not just sequestered in the corner, and the malignancy becomes prolific. These become important because as we understand the mechanisms that allow us to eliminate and provide equilibrium, and unfortunately be overwhelmed by escape, we can prime and work with the immune system to enhance those earlier phases so that we don't land in escape. So there, I've made you immunologists in the last 6 minutes. So on this note, as I shared with you before, we have this pre-malignant phase of myeloma called MGUS. There's even a step higher, something we call smoldering or asymptomatic myeloma, where there is more myeloma, but it's not hurting anybody yet. So we typically don't treat it. It's a bit of an unusual phenomenon in cancers, because most cancers, as soon as you identify the smallest amount of breast cancer, we need to eliminate it. But in myeloma, if we did that, we'd be treating 5% of the population. There's a lot of people, it's never going to hurt them. So we have to draw a line somewhere. And the red line here, as you see, is what indicates when someone has true active myeloma, which is on the basis, and I'm not going to go into much detail here, of what we call the CRAB criteria. When someone has a high calcium or renal or kidney problems, anemia or low red blood cell count, or some kind of bone disease, because that is where myeloma tends to proliferate, to damage those areas of the body. We have a few other biochemical markers and imaging markers that we've recently added as well. And part of this is the function that myeloma can be challenging to make, because people don't walk into the clinic and say, oh, I have this bad protein in my blood, doctor. Well, we don't screen for that. So people come in tired, they have bone pain, they have anemia or low red blood cell count. And for those of you who are involved in medicine, I mean, this is bread and butter for a family doctor. This is what we present to. I mean, there are people on the phone right now or on the Zoom call right now who are having back pain and are tired and maybe even anemic. I'm not making light of it. I'm just saying this is so common. No one immediately thinks of myeloma when these things happen. That's why the diagnosis can be challenging. Now, my last geeky slide, I promise, we're going to get very simple and clinical thereafter. This looks daunting, but I show this because this is kind of like a little history, a little walkthrough how we can treat myeloma. So the two purple cells here are myeloma. And the simple lesson from this slide is that myeloma is not just about the tumor, right? It's not just about the bad cells. It's about the environment in which it lives. This may be one of the most important scientific and biological points I can make to you today, which is to say that when we treat myeloma, when we think of myeloma, we can't just think of the tumor. We have to think of what the tumor does to its environment. So let me give you an analogy. Like I'm, as Celina mentioned, originally from Canada. I'm now a dual citizen. I'm pretty much a pacifist. So pardon me if I use a more aggressive analogy, but imagine an enemy invades the building across the street. So if I look across the street here, there's a Starbucks. No disrespect to Starbucks, okay? But let's say that the enemy invades the Starbucks and takes over the Starbucks. It's not just, excuse me, it's not just the fact that the enemy is in the Starbucks. It's that it takes over all of the features of the Starbucks. It now controls the Wi-Fi, the coffee machines, the barista machines, the all of the furniture there. Everything is now under the reign and control of that enemy. And that's kind of what myeloma does. Myeloma doesn't just occupy space by growing, like typically a tumor, let's say, think of a lung cancer, it occupies the space of the lung. I'm not making light of lung cancer, but it's pretty localized. It sends off some signals and messes with the body, but it's pretty localized. Myeloma really invades its environment and starts to take over the machinery of that environment. And this is what I try to show here in this picture, that there are stromal cells and T cells and natural killer cells and monocytes and dendritic cells. There's all these things, and then ridiculous number of pathways that guide this, that make it so intricate in the way that it affects its environment. It changes the machinery. So I don't know what a good example is. If it's Starbucks, now instead of making, I don't know, good coffee, it makes bad coffee. Maybe you think Starbucks makes bad coffee to begin with, but that's your prerogative. But now it's making unusual coffee, as it were, or it's doing it for its own purposes. So how do we take out these cells? Super simple, 3 ways. One way is we blow up the Starbucks, right? I mean, that's kind of a bit aggressive, but that's how we do it. We're going to find them, snuff them out, get them, just blow it up. And sometimes we do that with old school chemotherapy. We just give patients big whopping doses of chemotherapy to knock out the cells. That's one simple, rather aggressive way. One way is to say, you know what, the enemy, I know that they're hiding in the office in the back room. Maybe I could just target the office and try and knock out the enemy that way. So secondly, we have all these targeted therapies in cancer and in myeloma, where we target a pathway, some of the ridiculous number of pathways listed here, and we can specifically target the disease. Or thirdly, and this is where I'm going to come to at the end of my talk, is we can actually use the own environment to affect the cancer cell. Because the cancer cell embeds itself and is so secretly hiding in there, I can't get to the enemy. Maybe I hack into the building and now control the Wi-Fi. And now with control of the Wi-Fi, I can mess up the communication that the enemy has in that Starbucks. And that's basically what we do with immunotherapy. We regain control of the environment and turn the environment on the tumor, as it were, to help destroy the cells. So I'm going to show you later, for example, T cells. I can take T cells out of a patient and literally train them, because T cells are kind of trainable cancer cells, trainable immune cells, I'm sorry. We can train them to recognize a tumor, multiply them in the lab, and give them back to the patient. I know it sounds like you're watching an episode of Star Trek, but that's literally what happens when we do CAR T cell therapy. Chimeric antigen receptor T cells, we actually put a receptor onto the T cell to recognize the tumor, give them back to the patient after we've manufactured and multiplied them. So we take out 100, give them back 1,000,000, and that way it can attack the tumor. That's really the modern way of approaching cancer therapy. So in myeloma, I mean, this is just like a laundry list for most of you, and you don't even have to remember a word on this page, but we have pillars upon pillars of ways to treat cancer and treat myeloma, and I'll walk you through a little bit of this. Going back over 70 years, really, we started with old school drugs like urethane and melphalan, which we actually still use. With the 70s came bell bottoms and traditional chemo. And it wasn't until the 80s that we started doing autologous stem cell transplant. Transplant is really just a way to give very high doses of chemotherapy. The first true new drug in myeloma was right when I was entering the sphere of myeloma in 1999, really, where we used a drug called Thalidomide. Now, most of you were not even in utero to remember this, but this was a pretty tragic drug in the late 50s, early 60s that caused tremendous harm to women who were pregnant and indeed their children in utero when they were born with tragedies to their limbs because of the effect on the vascular system. But nonetheless, it had a resurgence in the late 90s and the early 2000s that it had activity in myeloma. And again, most of these drugs may mean nothing to you, but I'm just giving you a history of now we've introduced drugs that we use very regularly now called bortezomib and lenalidomide. And I put on the side some of the numbers that we started to see this drug work even in the most resistant diseases. I had the privilege of helping develop both carfilzomib and pomalidomide. And then in 2015, things really started to take off. In 1 year, we had 4 new drugs, as you can see here. And again, there's no test here to see if you can remember panobinostat, elotuzumab, daratumumab, ixazomib. These are all different, but daratumumab was a particularly important introduction that year because it was the first what we call monoclonal antibody, the first drug that really started to use the immune system, kind of like a mix of those 2 options I gave you, where it targets something on the outside of the myeloma cell, something called CD38, hooks onto it, and then triggers the immune system to come and destroy that cell. And that drug is now very prolifically used. Then most recently, and again, the acceleration continues, we've used these whole new classes of drugs, a drug called selinexor, a drug called isatuximab, which I personally had the privilege of developing. I led the first in human study on this over the last 10 years. I've been working with this drug, and it was very gratifying for me last year to see this drug finally get FDA approved after nurturing this little baby for so long. And for those of you who are involved in drug discovery and drug development, it is inordinately gratifying when you can start in the lab and eventually treat the first patient and eventually see it FDA approved, as I'll show you in just a moment. And now we have a whole series of new drugs, which I'm going to show you. First of all, let me just jump into this isatuximab. That this was the abstract, or the ultimate publication of this, that I had a privilege of leading this study where patients were the first to receive this drug. And as I mentioned, it is now FDA approved as of last year. But now we've added all these new drugs, and I'm not going to bore you with the names of all of them, but I want to focus for 1 minute on what we call CAR T-cell therapy, because this has been a modern marvel. We've seen it in leukemia, we've seen it in lymphoma, and now we're seeing it in myeloma, and we're seeing response rates that triple what we see routinely with any other drug. I mean, it's hard to not be melodramatic when describing this process, where the first time this was presented in our work and in our meetings, jaws of oncologists are dropped to the floor, which we don't typically do because we see just about everything, as you could imagine. And coming in the future, I think we can even get better at it. So just to quickly remind you of this CAR T-cell concept, what am I doing? And number one, I'm taking out of the patient these T cells, these cells that we know can be trained. And then I take them to the lab, and these next several steps are all outside of the patient, manufactured in a lab, where we enrich these cells, and by means of transduction, we actually, in a virus, we actually put onto the outside of the cell a receptor so that it can recognize something on the tumor. In this case, it's something called BCMA, or B-cell maturation antigen, that can recognize that on the tumor. And then we grow them, literally multiply them in the lab, and prepare them to then, number 6, as you can see here, give back to the patient. So it takes about a month to do so, a little over a month, and we give it back to the patient. And then now this CAR T cell, pictured in green, can bind the myeloma cell through this thing called BCMA, which is very prolifically expressed on the surface of myeloma cells. And it activates that T cell and can actually activate other things in the environment to help destroy the cell. This was published in the New England Journal a few months ago for the very first CAR T cell therapy in myeloma. It has a name that no one could pronounce. We call it ida-cel for short. We take the first 3 words, first 3 letters, the first word, and the last 3 letters of the second word that you can see there, and just call it ida-cel. And I won't go through much detail here, but I just want to notice that these are patients, this was given about 130 patients. These are patients that pretty much had everything we have for myeloma. They had 6 prior lines of therapy, which means basically they had life expectancy of less than 6 months because there was nothing left for them. And yet we saw a response rate here. The overall response rate of the whole cohort is 73%, but at the target dose of 81%. The best we have for these patients is maybe 20 to 25% with any other combination of drugs. And as I mentioned, we've even tripled that. And not only that, it lasts about 9 months, and in fact gives people an overall survival of nearly 2 years. In fact, we have a new CAR T cell therapy that may be even able to double this because we're getting better at it. I mean, this is hope for patients that otherwise we would never have had. Now, it comes with a challenge, and I won't get into much detail, but there is this thing called cytokine release syndrome. The immune system, it's tricky. You want to be careful how you play with someone's immune system. When we give the T cells back to patients, the body can react and say, hey, I'm not sure I like this. And it causes what we call CRS, or cytokine release syndrome, and the patient can get quite sick. But we are getting much better at managing it. And so they concluded that it has a 73% response rate, extraordinary depth of response, and most of the toxicities can be managed relatively well. This is fascinating because Myeloma is a kind of disease with time that gets more and more aggressive. That's kind of how most cancers are, as you know, but particularly in myeloma. So if we can prove something works that well late in the disease course, when it's very, very refractory to treatment, we really hope we can bring it earlier. And that's the goal of this next few years, to bring the treatment earlier into the disease course, because sadly, with each line of therapy, fewer and fewer patients are eligible for treatment. So a lot of my patients won't make it to CAR T because they die earlier in the disease course. But now, as we can bring it earlier, hopefully we can affect a difference. So for those who know a little bit about myeloma, this is kind of where we are with terms of treatments. I'm not going to go through this in any detail, but I put in red here the way we're changing everything. We're intensifying our treatments early on so that we can hopefully keep people in a deep and a durable remission for the longer term. What we do for patients earlier in their disease influence the long-term outcome. I'm going to make a quick few comments about other diagnostic sides, and then we'll go to the rest of the planet before I wrap up in about 20 minutes. So the other thing that has really changed, it's one thing to create great drugs, but it's another thing now to start imaging better. We know that over half of myeloma patients, probably more like 80% of myeloma patients, do have some kind of bone involvement for reasons we don't fully appreciate. These plasma cells that go rogue, they send out messages, and sometimes they send out themselves out of the bone marrow, the factory of our blood, and they start damaging bone in different places. And so in the old days, we used plain x-rays. And if I know you're not x-ray experts, maybe there's a radiologist of note in the crowd, but this looks pretty good to me. I mean, you can see someone standing there and you can see their spine along the back and down into their hip, into their tailbone. So with a plain x-ray, this looks pretty normal, but we've now developed better CT techniques where if we look at this exact same picture, that bone looked pretty intact, but we look at the second picture, there's a hole there. And that's what myeloma does. And we don't want to wait until someone breaks this bone and has horrible pain to detect it. So imaging becomes important. The other feature for those of you that are in the lab, and I know that some of you are into this sort of thing, so I'll just show you the picture in its entirety. Myeloma is a disease, as I mentioned, where bad proteins or antibodies are made. And we kind of test it pretty crudely now with something called a serum protein electrophoresis, or SPECT. I see the future of myeloma where we use this mass spec technique, where we can down to the Dalton identify the bad proteins in someone's blood. And this will be a game changer, I really think, for us in myeloma, being able to detect these earlier, more accurately quantify the disease. Because myeloma is not a disease where one test tells us the story. I tell my patients, unfortunately, myeloma is kind of like a crime scene. You can't just take one piece of evidence and make a conclusion. You need multiple pieces of evidence. And this is a very important one as we go through. Now, before I wrap up this section, to introduce us to the disparity section, I do want to make a comment about myeloma in African Americans, because this is unique. Myeloma is pretty rare, but it's common in the African American population. In fact, it's twice as common in African Americans as it is in Caucasians. And even though I've talked about all this great survival improvement that we've seen in myeloma, we have seen half of the survival improvement in African Americans that we have in Caucasians. That's not acceptable to me. And for many of us, of course. And so we really have to be serious about how we address this issue. As I mentioned earlier, the biology is a little bit different. They're diagnosed younger. They actually have a different genetic profile. I won't get into that in too much detail today. And sadly, a lot of the disparity in outcomes has to do with how African Americans access therapy. Although African Americans comprise nearly 20% of all myeloma patients, there are only about 5 to 6% of those on clinical trial. They're less likely to get transplants and what we call triplets, the drug combinations that work so well in myeloma. The point of hope here in number 5 is that we have shown through like VA studies and others, when African Americans and Caucasians are given the same treatments, the survival outcome can be just as good, if not actually better, in the African American population. So we have this hope and this recognition that if we do better, we can see much better outcomes. And that's what drives me and so many of us to do so much of this work. So this disparity is international. So in the role that I have, as Celina was kind to mention, I serve as the Chief Medical Officer of the International Myeloma Foundation. I've had the privilege of being involved in many global activities. I left Mayo about 3.5 years ago Joe can say to some that I didn't want to just treat rich people here in Schnottsdale, that I wanted to bring the benefit of this drug development of these activities to the planet. And one model of doing that, there's lots of ways of doing it, but one way that we have pursued is this notion of a collaborative network. And they're designed with many purposes. Sorry, this should say many purposes, not purpose. Many purposes in mind. And let me tell you about this concept of a network over these last 15 minutes. A network has the potential to benefit everyone. It can give access to these new drugs to patients. Most important, isn't it? That we care for our patients. It teaches physicians in these countries and gives them experience in clinical research, expertise with these new drugs, and even international prominence that they are able to collaborate with other investigators around the planet. The companies that make these drugs, and it has to be a collaborative effort, they can see greater penetration of their drugs, they can engage the providers and the patients. The countries themselves, who very often may not have the ability to fund many of these drugs and suffer the consequences of the health disparity within their countries, they can benefit from a network. And then, of course, the greater myeloma community, and part of the role I play in coordinating a lot of these efforts is really to try and strengthen the myeloma community, not just in the wealthy and resource-filled countries, but in many of these other places. So let's dive in a little bit more about this. So in many of these countries, here I've talked about this ridiculous number of drugs available to us and how it's changed how we treat myeloma and improved survival of myeloma. You know, I go to some of these countries. I'm going to be focusing today in Asia and a little bit of Latin America, but I also spend time in North Africa and the Middle East. We may have 15 drugs for myeloma here in the US. I go to some of these countries, they may have 2 or 3 max. So access is limited. Sometimes they get approved from a sort of their equivalent of the FDA in these different countries, but it doesn't mean it's accessible, because often it's very expensive and impossible for patients. And so when we look at our improving survival, what the potential is for myeloma, we go to some of these third world countries and we see abysmal survival outcomes. We know that clinical trials in many of these places are very limited. Some places have them, of course, but in comparison to the prolific nature of, you go to clinicaltrials.gov today, even in the US, you're going to see thousands of myeloma studies. That's not going to be the same in Paraguay or Costa Rica or Morocco. These countries aren't going to have that. And very often there's little advocacy and support for those patients going through it. There aren't a lot of not-for-profit or other organizations. We're spoiled here in this country with everything from the American Cancer Society, but obviously the International Myeloma Foundation, Leukemia Lymphoma Society. I mean, there's so many groups that try and support patients through this. So we can't fix that all in one shot, but building a research network with multiple countries working together has the potential to help this. Well, the physicians, many of these physicians that I interact with in these countries, they really want to participate in clinical research. There just isn't a resource for it. There isn't a budget for it. There isn't a research director. There isn't this beautiful, our hosts today are marvelous examples of an infrastructure that's built to support research. They may not have that. And often they feel that they're like observers. They just have to hear what other centers and places do, and they just follow the lead. They want to be involved, but they're also so busy clinically. It's hard to just add more to their plates. These people are seeing patients morning and night. So there's so much about research that has to be experienced. You can't just read the paper and know. And hopefully many of you are experiencing this in your degrees and in your work, that you've got to get hands-on experience to really understand this. And in my experience, these providers, these physicians in particular, and some of them are other advanced practitioners or pharmacists or others, they really want to get involved. And so we have a potential of giving them some kind of project that I'll explain in a moment. The companies themselves, they look at a lot of these countries and say, there's just no point. Why would I go to Paraguay? Why would I go to Ecuador? I mean, these are small countries, not a big market. Let's use the example of the US. No disrespect to, let's say, South Dakota, pretty small state. If you were a company, whether you're pharmaceutical or not, like any company, if you were like selling, I don't know, Oreo cookies If the whole of the United States was only South Dakota, you might not be that excited. But if South Dakota is part of 50 states, where you can do a distribution pattern and a sales pattern that covers the whole of the country, now all of a sudden it's very attractive. So as I say, Ecuador itself may be too small to really be a big enough market, but if it's part of a network where we can facilitate the regulatory process and assist with that so that multiple countries can be involved, now you have a network of 5, 10, 20 countries, where now the net size of that network is quite large, that is much more attractive. And so it allows a greater interest in being able to participate in a network and in clinical trials that the network will run. And so now drugs could be available to more patients, not just the entitled few. And so they can bring their experience of clinical trials internationally to more places. And again, networks have this opportunity. The countries themselves, I mean, they struggle with cancer care access. Obviously, there's a great burden of disease for many of these patients. Many of them end up having to be in hospital because they don't have access to outpatient-like treatments that we can use here. And this has the potential, if we could introduce some clinical research to these countries, to have a downstream effect and positively affect the health of the country, but even the economics of the country. Again, I don't want to get beyond myself. I'm just trying to give you the principle and the concept behind it. And then, of course, it has the opportunity to really benefit the whole of the myeloma community. We're all stronger when we work together. I know it sounds like the part of my talk where we all hold hands and sing Kumbaya, but the reality is that there is so much that we can do together that we wouldn't be able to do individually. And this really can help a greater overcoming of the equity disparity that we see in representation around the world, so that we don't have these higher and lower tiers of work, so that we can work together so much better, as the whole, of course, is greater than just the sum of its parts. So one network we created was the Asia Myeloma Network. This was started many years ago. This was one of the summits we had several years ago. And the concept was to introduce clinical trials into the Asian region. It was coordinated in one center at the center in the National University of Singapore. And through that, we had a management process to make sure all the data was secured. And then there are site agreements between multiple places. And I got to tell you, we have 8, we initially had 8, now we have 9 countries working together in this network who normally don't work together. We're talking about China, Japan, Taiwan, Malaysia, Singapore, as was mentioned, South Korea. I mean, there are many countries who you wouldn't think naturally are going to bind together and work together. Now, not every project, every trial, every activity has to be shared by the whole of the network, but it's offered to the whole of the network. And so they can work within the confines of what works best for them. So to just give you a flavor, we've done several clinical trials now through this network. And these aren't clinical trials that are necessarily answering dire scientific questions. This is the focus now, that they're interested in giving access to these patients. So for those of you who've done clinical trials here in the US, we often do what we call EAPs, or expanded access programs, or clinical trials. They're kind of built on that notion. Let's say this drug pomalidomide, or CAR, standing for carfilzomib, is really not available in many of these countries. We can introduce a simple trial to bring access to the drug, so the docs get used to it, the patient gets access to it, we gain information. Is there a difference in the response rate in Japan versus China? Is there a difference in the toxicity levels? Do we have to adjust differently in one country over the other based on the individuals and the patients that are participating in the study? And so we have many studies like this. I'm just showing you lists, some of these drugs you've never heard of, but these are drugs that we've now been able to introduce over the last several years of many of these new drugs. But it's not just the trial. It becomes a partnership. People have to feel like they're working together. So we do a lot of physician training to help them understand it. This is a talk that I gave in Guangzhou a few years back, where we had a summit to bring people together to discuss what's the state of the union of myeloma. How are we going to approach this? They want to understand the disease and the drugs better to be able to use them, not just for the docs, but for patients. Some of these countries never really had large patient seminars for this disease in particular, for myeloma. And so in this case, this was in Korea, in Seoul. We had large patient meetings. Of course, this is before the days of COVID, although you do see a few masks there, where we could educate patients as well, so that they could be involved and engaged in the process. More recently, I had the privilege of helping to create, and we're just still in the formative stages of the Latin American Myeloma Network. So following the model of what happened in Asia, we're now bringing it to Latin America so we can have dialogue between these countries. And if you know much about Latin America, there's tremendous disparity there, tremendous difference. You got a huge country in Brazil, Portuguese speaking, and many mostly smaller countries, although a few big ones like Argentina, that are Spanish speaking. And often there's discord between the two. So we wanted to enhance the activities within the Spanish speaking countries that have limited options and resources, and have them working together alongside the Brazilians. And I've seen over these last few years building trust, and it takes time. None of this happens overnight. I've been going there for 7-8 years to try and build this kind of trust, that we can indeed create a network. So just giving an example, this is the executive of the network of physicians that I'm working with in Latin America, 3 of which are from Brazil and 4 of which are from other countries, including, as you can see, Argentina and Chile and Uruguay. So as I get towards the end here, let me give you an analogy to try and bring much of this together. Imagine, sadly, a very poor village, Africa or India or somewhere where unfortunately the resources are very limited. And I said to you, your goal, your objective is to improve the survival of people within that village. What are you going to do? Well, several things come to mind, and I've listed 5 things on this graphic to help us see that. For one, we need to build a well because they need a source of clean water. In the middle, we need a maternal health program to help mothers as they care for these little ones. We need a schooling system. We need an opportunity to have waste dispensed with appropriately and have restrooms that are not connected to the drinking water, as sadly happens in many of these places. And we clearly need some kind of vaccination program. Now, if I send you to one of these tiny villages in one of these countries, you can't just do that overnight. You might have to say, look, in this village, they really need the well first. But in this village, they absolutely need the vaccination program first. And so you focus in an area. So I don't want to make it sound like this is some cookie cutter approach as we go. We have to learn, just like every patient is different, every community is different. We have to say, you know, when I'm doing some of this work, we say in Chile, for example, more people are rural. So maybe we tailor a treatment program that has more oral therapy. Whereas in Uruguay, the majority of people are close enough to the capital of Montevideo that maybe we do it with an intravenous process. That's how we have to think a little bit differently in drug development and indeed in clinical trials and ultimately in access for patients. Just because it worked in one country doesn't mean it can work in another country. And so it's so important as we go through to think about this. But I hope that even just sharing this concept and this idea with you induces a little bit more passion for recognizing those that are suffering more than we are, for those that have less than what we have. So some key messages I've learned through this process of network creation. Every country is different, as I mentioned, it has to be respected. Collaboration is absolutely fundamental. That's why I listed it by all the parties involved. No one party can just run the show. Physician ownership in these countries has to be critical. We can't pretend like I'm coming in, descending with my cape, I'm going to be the savior of this place. No. They have to own it. I'm just a facilitator. And ego in that way has to take a backseat. There's a lot of ego in medicine, as you all know. Regulatory issues are a pain in the tuchus, but they can be overcome. We can find ways to do it. This does take money, obviously. This is not done cheap, but it can be done. And when you think of the investment and what it can actually return, it's very much worth it. And of course, we have to be driven by patient need. That's what motivates me every day to come to this clinic, to care for our patients, obviously, is for their benefit. So as I look over the years of myeloma therapy, what a change. In red here is what we've been using for a long time. In green, we've just added. And in purple on the bottom, I show you that there is a whole lot more to come. I haven't even started to tell you about some of the newer therapies. I'm just opening a clinical trial here this week, where instead of a T cell that we collect from a patient and manufacture and give back, I can give a drug that has two arms. One arm hooks onto the tumor and the other arm reaches out. We've done this with T cells, but now we can do it even with natural killer cells, as I showed you in that first diagram. And I can hook that natural killer cell right into the tumor, as it were, so it can help destroy the tumor. So I never have to put the patient through that whole collecting T cell notion and waiting 4 weeks. I can just give them a drug directly that initiates their immune system to try and destroy the tumor. These are the kinds of things that I really think are going to change the future of what we do in myeloma. Let me close with this slide, because this is one of my favorite slides that I always like to give a balance to what we've said. Drug development is great. I can tell you all day long about fancy schmancy drugs. But let me tell you about this study. This is actually a study done in solid tumor, not in myeloma. But this was a study that randomized patients to receiving an iPad to communicate more clearly with their healthcare team versus kind of the standard of care, which, if you know much about this, the patient calls, leaves a message, gets a call back from a nurse later. And they were hoping to improve quality of life by doing this, and they did. But what they didn't fully anticipate is they actually improved quantity of life. Patients that communicated, and physicians and healthcare teams that communicated better with their patients, resulted in longer survival of their patients. So as I say, I can tell you all day long about fancy schmancy drugs. At the end of the day, maintaining communication skills, being able to communicate with our colleagues and our patients is going to enhance what we do in the future. So I'm going to come to an end here. Here's just a little picture of, I've given you a couple of spots, but really all over the planet doing great work that I'm blessed to be a part of to try and improve the outcomes of patients with multiple myeloma. It's been an absolute joy to share this with you. I hope it's been helpful to you and of interest to you. At this point, I am going to stop sharing my slide and turn it back to you, Celina, as questions may or may not have come in. I'm more than happy to try and answer them. I hope this has been of interest to those who have listened in today. Thank you again for inviting me to this amazing conference.
Well, thank you so much, Joe, for participating. It was a fantastic story that you told us today, and really inspiring. I think that's pretty amazing. So I'm really inspired by your talk. So I have a question here from Souvenir Tachado. Can a second dose of CAR T cells work when the first one fails?
Yeah, great question. Thank you for this marvelous question, actually. So one of the greatest benefits of CAR T cell therapy from a patient's perspective, I had a patient telling me this just a few weeks ago, saying, you know, doc, compared to getting these drugs given to me week after week after week, one nice thing about CAR T cell therapy is that you kind of get this big whopping dose, but then you have nothing afterwards. And so this patient of mine had been off all treatment for 2.5 years in remission, which is a big deal in myeloma because most of our patients have to stay on some form of continuous therapy. But inevitably, there are patients where, as we saw, I mean, CAR T cell therapy isn't curative, where the disease is going to relapse. And so there's two ways to answer this great question. One is we tend not to immediately give another set of T cells, although we are looking into whether or not a booster, I know that's a big word right now, a booster of T cells may be helpful. But we have now started a process where I think of a patient of mine who 3 years ago, we gave him a CAR T cell on clinical trial, and his disease was in remission for nearly 3 years. Unfortunately, he's relapsed. And now actually we can give him another CAR T cell with a similar target. Sometimes that BCMA target is retained, although we even have new targets. So the second and simple way to answer your question is yes, there is an opportunity. It isn't just the one time and that's it. You only get one kick at the can to use T cells. I do think that as we explore new targets, as we explore, as I mentioned, natural killer cells, we may likely be able to give people different ways of doing that. But even right now, we have patients that have had 2 different CAR T cells.
Thanks. I just want to also remind the hosts and the speakers that you don't have access to the Q&A, so you can just unmute yourself, put yourself in a video and ask questions if you have them. We still have time for a couple more questions. I'm going to ask one. I'm very fascinated by genetics in all diseases, and I noticed that you said that there is a genetic component that you're not going to talk about today. But can you talk just a little bit about, is that the reason why Hispanics and Blacks have African Americans have an increased risk or have more incidence of myeloma is genetically driven?
Yeah, so when we think of myeloma, great question, Celina, of course. When we think of myeloma, we don't think of it necessarily as a genetic disease, meaning we although there is a small contingent of patients that have a family member with it,
okay,
it's not like some other cancers where we clearly see a strong familiar pattern. So over 98% of patients have no one in their family that we can really identify as connected. That being said, there's two ways of thinking about it. First of all, from someone's inherent genetics, of course, we definitely see different patterns. So with those who have African ancestry, for example, not only do we see it more commonly, we then secondly see that the cytogenetics of the tumor cells, so not someone's inherent genetics, but the way that the tumors manifest itself, we see a different pattern of genetics when we compare one group to another. So for example, there's a translocation 1114, which we know in myeloma tends to be a relatively good prognostic marker, meaning when people have it, they tend to have better survival with the disease. That's more common in African Americans. And that may be one of the reasons why, when given equal access to therapy, they can have better outcomes. By contrast, one of the cytogenetic features we look for that is unfortunately an adverse prognostic factor in myeloma, something called the 17p deletion, where the P arm of 17p is missing because we miss the gene, the p53 gene. That's associated with a poor prognostic factor. That's actually less common in the African American population. So we're now trying to understand the same thing within the Hispanic population, the Asian population, and others. So the short answer is yes, there is a connection. We're still trying to understand it, but we don't tend to think of it as a familial condition in most people.
Okay. Does anyone, we don't have any questions from the participants. Does anyone, oh, there is one question here from Claudia Whitehead. This is amazing and offers so much hope to everyone with the advancement of these therapies. As you work internationally, how do you work with the local communities? As a former Peace Corps volunteer, I think working with Peace Corps volunteers could be a great partner to work with the local communities.
Mm-hmm. Well, thanks so much for the kind words, Claudia. It is really, I hope it provides hope. So at least one person felt that today. So I appreciate it. Everyone comes from their angle. I come as a physician in the medical angle. So most of my connection there, so it's sort of my nirvana trip to one of these places is I will meet with the docs and the individuals that would be involved in clinical research. But I also like to meet with the health authority if at all possible. Obviously, I often even meet with patient groups. And then because I love mentorship, I try to meet with the junior physicians or junior hematologists and oncologists and work with them. So it goes back, and I think your point is very well taken about what you do in the Peace Corps, is that you don't sort of come in and say, this is how you do it, you'll do it, you'll like it, right? You work within the confines of that community. This is even the work that I do within the African American community. I don't pretend to be some savior and show up with the answers. I work within that community and build partnerships with the medical and the non-medical aspects of the African American community or any other community, so that this is embedded within the community to understand what we can do to better people's outcomes. So you make, I think, a point that is very critical for all of us to understand, is that we don't just sort of come to the rescue. And we look at any efforts that have helped third world countries. It's not that we just come in from the outside. We have to leverage the whole infrastructure of that own community to make it better for themselves. And frankly, that's the only way it'll be sustainable.
Thanks. Any questions from the panelists or the hosts? I'm going to ask one final question, I guess. Do you think that the device that allows the patients to communicate directly with, and now I have to wait for a phone call because I know a friend of mine just had cancer and he was waiting for these calls and it was so stressful. Do you think it's related to the stress effect on the immune system? That
I mean,
there's stress if you can communicate immediately about your.
Yeah, this is a whole growing area. And I see that Bill's got a question too, so I'll be
quick.
He does. But this is a huge growing area of cancer care. I included that study because it set the precedent to say that the communication can. Now we deepen that communication, just like you said, we can transmit health information, and we have to obviously be very careful with, HIPAA rules and confidentiality and so on. But we have ways of, especially now in our virtual world, I can get to see some patients virtually today where we can transmit, temperature and blood pressure and heart rate and these kinds of things. Now we even have sometimes some studies I'm involved with have patients that have a smartwatch. And so that transmits information. I really do think that is going to be a big part of what we're doing in the future, because it's one thing to see the patient on that one day. It's another thing to have more data, sort of big data collected in advance.
Okay, Bill, you're up.
Okay, thanks. I've lost the ability to show my video, so I'll just ask blindly here. So I have two questions for you. One is more technical and one is more philosophical, I guess. The technical one is, so you said that there are advances in treating this cytokine storm that comes with the CAR T treatment. But I'm just wondering how that is being dealt with, since you mentioned it. And the second is the CAR T treatments are really expensive, as I understand it. And so how is the expense being dealt with in the community? Is everybody getting access? Getting access to third world, it must be extremely challenging, but even in the US.
Two great questions, Bill. Thank you. I'll answer them quickly because I know our time's going quickly, Celina. One, identifying CRS is there your face appears. Is not just the intervention, but it's the earlier recognition. So, for example, through the IMF, we have what's called the International Myeloma Working Group, and we have an immunotherapy subgroup, and we meet regularly so that we can share best practices, so that we can have very clear criteria to identify what is CRS and what isn't, because we've learned the earlier you intervene, the better. And part of that intervention are strategies. Sometimes we use steroids. There's a bit of a national shortage, but we use a drug called tocilizumab, which is a drug that can help significantly reduce the impact of CRS. So part of it is drug intervention, but part of it is earlier recognition. The cost issue with CAR T is a very legitimate question and a huge one, and one that's being evaluated, and ICER and others are trying to look at this. The short version is that obviously, we hope with time that things get cheaper, right? Cheaper phones over time, cheaper technologies over time. But we have to factor in the difference in this is the depth and the duration of response and the lack of treatment needed thereafter. So we can't just look at the initial sticker. It's kind of like saying I got to buy an electric car, maybe a hybrid car, maybe more expensive up front, but then the maintenance fee thereafter is significantly lower. That's the kind of way we have to look at it. But yes, I mean, thinking about bringing CAR T to some of these countries is just a pipe dream right now. But we hope, and maybe I'll end with this, that the way I've described CAR T is the way we're doing it now. There is a hope that with time, instead of having to take a patient's T cells out, I can actually pre-manufacture T cells and just give them to the patient. So almost like giving them a drug. And that, of course, would radically change the price of being able to do it.
Yeah, and I guess it also might just be a temporary solution to the problem based on the new advances coming along.
Correct. Thank you again for great questions, and I'll turn it back to you, Celina, as our time is quickly.
Thank you, Joe, again, and really appreciate you taking time and giving this amazing talk today. So we now move to our second speaker. I'm also super delighted to introduce you, our second speaker, Dr. Chris Glynbotsky. We're incredibly lucky to have this renowned researcher in cardiovascular disease recently joined the University of Arizona from the San Diego State University Heart Institute. He's currently professor of the Department of Internal Medicine. He's a professor, he's director of the Translational Cardiovascular Research Center and associate dean for research at the College of Medicine in the Phoenix campus. Dr. Glembotski obtained his bachelor's degree in biochemistry from the California Polytechnic State University at San Luis Bishop in California, and his PhD in biochemistry from the University of California, Los Angeles. He then trained as a postdoctoral fellow in molecular physiology at the University of Colorado Health Science Center in Denver, and then moved to his first faculty position at the Paramount School of Medicine in the University of Pennsylvania in Philadelphia, before relocating to San Diego to become the director of the SDSU Heart Institute. Dr. Glembotski has extensive research background and garnered more than 35 million in grants during his time at SDSU. He's been continuously funded by the NIH since he started his own lab in 1983. His research centers around the area of molecular cardiology with a focus on identifying new therapies for treating ischemic and hypertrophic cardiomyopathies. The title of his talk today is Proteostasis as a Therapeutic Target for Treating Heart Disease. Welcome, Chris, please take over.
Well, Celina, thank you so much for such a great introduction. I appreciate you presenting some of my background of where I've been before, and it's been our research in various aspects of heart disease that has led us to where we are today. And as I reflect back, it seems like every bit of that research moved us towards therapeutic. And so we had to discover a lot on the way, and hopefully that's what I'll tell you about today. Hopefully you'll find it an interesting story. We have many other stories, but I thought I would focus on this one today. So thanks for the invitation. It's a real honor to present to this interesting group of participants and the panel. And so I just thought I would start and tell you about our research overview for our lab, just to put in perspective what I'm going to talk to you about. Our goal is to make the heart more resilient to disease. So that's our high level goal. Our objective is to enhance the heart's own natural disease defense mechanisms. And so our main approach is to discover those mechanisms and use typical molecular biology techniques like targeted gene expression and deletion in usually mouse models is what I'm going to talk about today, where we can mimic heart disease, such as myocardial infarction and pressure overload are our two main heart disease models. And we really focus on the heart itself and the myocardium, the muscle cells, cardiac myocytes. And in so doing, look at cardiac structure and function in the mouse models where we've done these maneuvers to see whether the genes of interest that we've altered have the predicted effects on making the disease more resistant to heart disease. And most of the heart disease that I'll be talking about today is damage due to myocardial infarction, which is a big problem because, as most of you know, the heart does not regenerate myocardium quickly enough to really repair itself with contractile functional muscle tissue. So this leads to a disease that we study a lot in lab called heart failure. So today, though, I'm going to tell you about one of our therapeutic approaches involving small molecule drug candidates. We have several other approaches, including gene therapy, as well as peptide and protein transduction approaches. But today I'm going to focus on the small molecule drug candidates. So why do we have this drawing of the mouse here that looks like he's celebrating his birthday? Well, I'm going to talk about mouse models today, but also we're celebrating our first year anniversary here in Phoenix. We moved in September of 2020, and we initiated the University of Arizona College of Medicine Phoenix Translational Cardiovascular Research Center. Sometimes we abbreviate this as TCRC. So this has been a month-long, maybe it'll turn into a year-long celebration of great opportunities that we've gotten since moving here, including interacting with Celina and others in the ACDD. So I want to focus on proteostasis and just give you a brief overview here in this diagram of what we call a proteostasis network. This is one of the simplest diagrams that depicts the life cycle of a protein, from amino acids to being translated, to folding, to having its native state and function, to eventually being targeted for degradation, usually due to obsolescence or signal regulation, turned into peptides and amino acids, and then we start the cycle all over again. So although this is simple, maintaining a balance of all of these elements is what proteostasis is all about. And that balance is required to maintain our proteome integrity and therefore optimize cell function and adjust to different situations that cells, tissues, and organs might meet on a daily basis, let alone during pathologies. So the focus on proteostasis that I want to talk about today is in the endoplasmic reticulum. And the ER became our focus because it's a specialized place where proteins get made, folded, and then targeted to their various locations where they can become membrane proteins, secreted proteins, and others. So this is a picture of the ER, just a diagram I drew that shows some ER luminal proteins and membrane proteins. And they get to the ER by co-translational translocation, not shown here. And some of them go to the Golgi and then eventually to secretion or to the plasma membrane. And this is a very delicate process in all cells, but especially those cells that have to make important membrane and secreted proteins. And cardiac myocytes, proteostasis in the ER is critical for the synthesis and folding of many important proteins, including hormones, growth factors, receptors, contractile calcium handling proteins, and stem cell helping factors, just to mention a few. So these were the underlying compelling reasons why we focused on the ER. And when we started our studies, really the endoplasmic reticulum in terms of protein synthesis and proteostasis had gone unrecognized and unstudied in the heart. So this led us to a better understanding of the unfolded protein response, which was studied in immune cells and cancer cells before we started studying in the heart, maybe 20 years or so ago. And when there is a perturbation of the ER folding environment, which can happen when there's a lack of oxygen or when there's other metabolic changes, it's quite easy to perturb this environment. Commonly, there's a condition called ER stress, which is a result of ER protein misfolding. And there's three ER sensors of this ER protein misfolding, ATF6, IRE1, and PERK. I'm not going to talk in detail about really any of them today, but suffice it to say, one of the things that all three of these sensors do is reprogram the genetics of cells in order to mount a resilience or response to this ER protein misfolding problem. So our overlying concept that originally drove our interest in the UPR in the heart starts with cancer and the discovery by others many years ago when the UPR was first discovered that tumors have a really robust unfolded protein response. And as a matter of fact, it's adaptive in tumors and it correlates with their metastatic potential. So what we wondered was, what about the heart? And might the heart have not a robust, maybe a weak unfolded protein response, and maybe we could boost it to protect the heart? So sort of a case where in the heart, your therapeutic strategy would be to activate adaptive ER stress response, whereas in tumors, you might want to inhibit. And when we embarked on this, as I said, nothing was known about this pathway in the heart at all. So the pathology setting I'm going to discuss today is called ischemia reperfusion, or abbreviated IR, in the heart. And this diagram depicts all the elements of this, where a clogged coronary artery can reduce blood flow, causing ischemia. And then oftentimes this is opened clinically by placement of a stent or other modalities. And then the stent causes blood flow to resume. And so we call that reperfusion. And in terms of the damage that's caused during these events, there's this diagram here that depicts theoretically if there's an infarct that is, say, 70% of maximum due to permanent occlusion, that would be myocardial ischemia in the absence of reperfusion. We could theoretically set that infarct size at 70% of the total heart cross-section here, and the infarct is the white tissue. And it's important to get the stent and to reperfuse the tissue because that reduces infarct size by 40%, all the way down here to a total of 30. But what happens is there's a generation of ROS, reactive oxygen species, during reperfusion, and that causes even more damage, all the way down here to probably accounting for another 25% of the infarct size. So if we could prevent this reperfusion injury, then we could optimize, increase or decrease the size of infarcts and increase myocardial function and patient survival. So we embarked on studies of IR in mouse models, and I'm going to explain to you ex vivo, where we can take the heart out of the mouse, genetically modified or treated mouse, and we can study its function. And that's shown here in this diagram, where we keep the heart going on this kind of a heart-lung machine, and just focus here on the chamber where we show a magnified view where we put in a balloon in the left ventricle, the main pumping chamber of the heart, with a pressure transducer, and it measures cardiac contractility with each heart rate. The output of that transducer as a function of time looks a little like this, where each of these peaks and valleys represents a heartbeat, and the area under those curves represents left ventricular developed pressure, abbreviated LVDP. So in this model, we can shut off the flow to the heart, and this is global ischemia, and guess what? The heart stops beating. So it's a pretty major crisis, and then we do this for some period of time so that when we resume the flow, the heart will start beating, but there's been some damage, and then the amount that the beating comes back is the recovery, almost like a patient who's getting a stent after an ischemic myocardial infarction. So here you see the beating has come back, but it's much less than it was in the beginning. And if we take lots of these data points and plot them together from primary data to the aggregate or cumulative data, a graph would look something like this for LVDP as a function of time, where we introduce this ischemia, the heart stops beating, and then we reperfuse, and you see there's a return to function, but it's not as good as it was when we started the ischemia. So we decided to focus on just one branch of the ER stress response, or unfolded protein response, and depicted by ATF6. It's been a major focus for maybe last 10 years or so. And just to remind you, it is a transcription factor that reprograms cells to make more ER chaperones and other proteins necessary to amend ER protein folding and rescue the unfolded protein response. Therefore, the cells would survive. So we started looking into the function of ATF6 in the hearts of mice, and we devised a way to delete ATF6 only in cardiac myocytes of our mice. So we call this conditional deletion of ATF6. To see what the functional consequences were, we chose the ex vivo experiment procedure I just showed you and measured heart function after a 30-minute ischemia and then followed by reperfusion. And this is what our graph looked like of developed left ventricular developed pressure as a function of time. And as I told you, with ischemia, the heart stops, and with reperfusion, the heart returns, and wild type mouse hearts returned here to about 50%. But if they didn't have ATF6, these hearts returned to about half that, or maybe 20%. We showed that we could restore the resilience of the heart by using a gene therapy approach to replace the deleted ATF6. I'm not showing you here. And so this showed that this was an ATF6 dependent resilience of the heart to this ischemic damage. So we can also take the heart after we're done with this and make sections and measure the infarct size of each of these sections and put them back together into the form of a graph. And you can see the infarct size, if we knock out ATF6, is much greater than in wild type mice, demonstrating the importance of endogenous ATF6 in making the heart in mice resistant to the damages of ischemia. We can do similar experiments in vivo, where we do an ligation of one of the coronary arteries for a short period of time and then open it up like a reperfusion event and measure infarct size as well as plasma levels of cardiac damage markers after reperfusion. And the infarct size in the ATF6 knockout mice is much greater in vivo like it was ex vivo. And then the release of markers of damage from the heart, like cardiac troponin I, which is used in the clinic, was also increased. So I wanted to remind you, I didn't actually say this, but ATF6 is expressed in every cell in our body, including if there's cancer cells, cancer cells. So that's why we used cardiac myocyte-specific knockouts here to prove that what we were visualizing was by virtue of ATF6 in cardiac myocytes. So ATF6 is cardioprotective, and what is the mechanism? So we use lots of strategies like RNA-seq and proteomics and found two major groups of ATF6 regulated genes in the mouse heart, known or canonical ER stress inducible proteins, which we fully expected, and unknown ER stress inducible proteins, some targeted to the ER, some to other locations, which were very interesting to us, including a group of antioxidant genes. And the one that we focused on and then we found is induced by ATF6 is the reactive oxygen species scavenger catalase, which was very surprising to us to find that it's an ER stress inducible and ATF6 inducible gene in cardiac myocytes. And interestingly, there's catalase goes down in the heart as a function of age, at least in mice. And so restoring catalase in these animals or inducing it with ATF6 has a protective effect by decreasing reactive oxygen species during reperfusion. And this is how it decreases, ATF6 decreases injury. So now we wanted to go into kind of a therapeutic approach. Now I want to remind you here that this ischemia reperfusion not only activates ATF6, but all three branches of the ER stress response as shown here. But what we wondered is, can ATF6 be selectively activated by a small molecule drug candidate? And can this activation boost the protective effects of ATF6, endogenous ATF6 during ischemia reperfusion? So this is the last part of the talk that's related to drug development. And so we embarked on a collaborative study with Luke Weissman's lab at the Scripps Research Institute in La Jolla. It's a chemical biology lab where their expertise is screening for various small molecule compounds, and they focus on ER stress and ATF6, albeit not in the heart. They look more in cultured cells. So that collaboration worked out really well, and we discovered a number of small molecule activators of endogenous ATF6 after screening their library, which is 800,000 compounds in 293 cells. And the one I'm going to talk to you about today is called compound 147. And this is the structure of the compound, and we have a control compound, which is only slightly different than our test compound, which we use in all of our studies. And these are cultured cardiac myocytes treated with either the control or the compound, which we tested in cardiac myocytes. And you can see all alone, it's able to induce the expression of ATF6 target genes, immunostained here in green. And this target gene was the ER chaperone GRP78. Mimics what happens when we stimulate the cells with simulated ischemia reperfusion. So we were pretty excited about trying this in our animal models. And so the first thing we did was to see whether it selectively activated only one branch of the unfolded protein response. And sure enough, as anticipated based on the screen, it activated only the ATF6 branch and neither of the other two branches. And this was important to us because we wanted to capitalize on the mostly adaptive effects of the ATF6 branch, whereas the other two branches can be a mixture of adaptive and maladaptive. So we wanted to focus the candidate on ATF6. So we went to the ex vivo heart again, and we said, what happens if we treat these hearts with this compound 147? And you can see during reperfusion, wild type mice recover in this experiment to about 50%. But if they're pre-treated with compound 147, they recover a great deal better. In this experiment, the mice were injected several days before the hearts were obtained for this experiment. If we used ATF6, mice where ATF6 is knocked out in cardiac myocytes, we got a very different picture as shown here. So it didn't matter whether they were treated with control or 147 beforehand. If there was no ATF6, there was a very poor recovery, similar for both in response to reperfusion. and demonstrating the ATF6 requirement for the adaptive and protective effects of 147 in the heart. So we can do this using various model systems, and we decided to take it to an acute myocardial infarct system that mimics what happens in people when they have a myocardial infarction. Maybe within a short period of time, let's say half an hour, they'll get a stent or go to the cath lab and be treated some other way, and then reperfusion happens. And so we decided to treat the animals just during reperfusion with either the control or the test compound, 147, because that's the clinically relevant scenario that was important to mimic. So what we found out was that the 147 treatment decreased the infarct size when animals were examined either 24 hours or 7 days later, and also decreased the plasma levels of the marker of cardiac damage. So remember, like ATF6 is in all cells, 147 is a small molecule and goes all over to all the tissues. And so we were able to try it in other models of ischemia reperfusion, including the kidney, shown here, where a renal artery is tied off and then opened, where renal infarct is greatly reduced, as is a plasma marker of renal function, in this case, creatinine. And then the other thing we became interested in was the brain. We found that compound 147 passes the blood-brain barrier. And so we want to test it in a ischemia model of stroke. And when we did that, you can see there's much less damage in 147 treated animals, as shown by decreased infarct size in a stroke model. And the neurological score, in this case, the lower the score, the better the function. And this is a physical activity of neurological function in these mice. So we're very excited by compounds like 147 and look forward to our future of taking this into larger animal studies, and hopefully soon maybe to clinical trials for compounds like this, maybe some to even be discovered in the future. So how does 147 work? So it turns out ATF6 is an ER-tethered protein that has an ER luminal domain that senses ER protein misfolding and translocates to the Golgi, where there's a protease that cleaves ATF6, and the cytosolic domain has a nuclear translocation signal, goes to the nucleus, and regulates the ATF6 gene program. Well, 147, it turns out, mimics unfolded protein accumulation in the ER, but only for ATF6. And when it mimics this, it stimulates ATF6 to relocate to the Golgi and otherwise go through the same activation it would by the normal unfolded protein response to induce genes that reprogram proteostasis to reduce myocyte death and provide us with cardioprotection. So we published these studies in Nature Communications, and one of the main people who did almost all of the experiments I just showed you today was Eric Blackwood with the team in our San Diego lab, shown here. And then the discovery of how ATF6 works, its mechanism of action, was done in collaboration with the Luke Weissman and Jeff Kelly labs at TSRI in La Jolla, and Ryan Paxman and Lars Platt were the primary investigators on that experiment. So this is a summary of how we think of our drug development pipeline in terms of proteostasis as a therapeutic target for treating heart disease. And we have these three main research models that we think about: cardiac biology, and I showed you something from each of these models today, where we use mice and cultured cells to discover those pathways in hearts that make them resilient to heart disease. Chemical biology to discover, for example, small molecule regulators of those same pathways. And then examination in models of cardiac pathology, initially in mice, later in larger animals, as is necessary for moving drug candidates down the development pipeline. And so this is how we think about our overview of how the lab functions here in Phoenix. So we're happy that we made the move because it made a lot of these elements of these modules possible. And the translational aspects of our research were majorly positively impacted by our move. And for example, being able to talk to a group like you are today. So I wanted to recognize the people in our Translational Cardiovascular Research Center. Eric Blackwood, who did most of the studies I talked about today with his team, Lauren and Lena and Donna. Donna stayed in San Diego, but Lauren and Lena moved with us to Phoenix. And then our other researchers here in the Cardiovascular Center, Drs. Derudkar and Jacoby, who are new faculty recruitments, been here for 6 months. And Bernie Sadowskis is, along with Tina Allen, our important administrative assistants. And then our other collaborators that I've mentioned, some more than others today, Luke Weissman from TSRI, and on the pig studies, Steve Goldman from ComT, David Leffer from Louisiana State University, and Gerald Churko. I didn't talk much about stem cells today, but he's a big collaborator of ours at UA ComT when it comes to our cardiac stem cells. So I thank you for your attention and look forward to if you have any questions.
Great. Thank you so much, Chris. It was really fantastic talk. I mean, the UPR is just really fascinating, and so many, people are working on this, particularly for neurodegenerative diseases and the other branches, of course. And I actually had written down, I was going to ask you about ATF6 in stroke, and then you came around and said that exactly that works in stroke. So really fantastic opportunity here for treating stroke, which, there's not very many therapies for it. So I do not see any questions from the panelists, from the audience, the panelists. Bill, go ahead. Bill is.
Oh, yeah, Chris.
Hi, Bill. By the way, I was going to compliment you on your background. It's really a great one. I
wish we were all there.
Yeah,
in Colorado. Well, thanks, Chris. That was really a lovely talk. I have a number of questions. I'll throw a couple out here for you to act on. The first one really is in your drug screen. Looks like you found some sort of a reductant and an activator at the same time. That must have been a very difficult screen to set up. Activators are hard to find to start with, so maybe you could tell us a little bit about how that screen came together.
So the screen, as you might guess, it's a cell-based screen. And as you might guess, it's based on, since ATF6 is a transcription factor, a engineered promoter that's driving a marker enzyme like luciferase or others, in this case, luciferase. And so it's a high throughput screen, and that's the primary screen. And that promoter element was engineered as carefully as possible to be specific for ATF6 and none of the other transcription factors that can arise during activation. And actually, I didn't tell you, but as a result or a byproduct, the screen can be used to look for those transcription factor activators as well. We have some that are IRE1 activators, for example, but I didn't talk about those today. And then the secondary part of the screen was to do a transcriptome profiling and then look to make sure, look at each of the near final compounds and look at the RNA-seq pattern and see if which one looks the closest to a typical ATF6 specific transcriptional induction landscape. So we focused on that. And then, of course, in there somewhere was the compound can be non-death causing. So we didn't look necessarily for survival with the screen, but for at least it doesn't cause death.
Yeah, So if I could just have one follow-up, it looks like we're getting a bunch of questions in the.
Yes, we do. Go ahead.
So the follow-up question is, what do you see the therapeutic window for these compounds being? Because on your ex vivo model, you had about 30 minutes to make a difference.
Yeah, so what I mentioned on the ex vivo was they were injected with the compound a day or two before we took the hearts and used them ex vivo. So the idea there is genes are going to be induced, and they're still induced by the time we put it on the heart apparatus. And so the gene program has already been reformatted for protection. The therapeutic window, so we did some initial pharmacodynamics, and as you might guess, the compound is cleared very, very quickly. Within 30 minutes, the half-life is, 30 minutes or shorter in plasma. However, what happens is the compound is actually a pro-compound. It goes to the ER, and then it's worked on by a cytochrome P450 enzyme located just in the ER. None of the mitochondrial or others will do that, just in the ER, and converts it to the actual compound. And what the compound does is it inhibits protein disulfide isomerases in the lumen of the ER that are the ones that keep ATF6 in that oxidative form and retained in the ER. So when those disomerases are inhibited, it kind of releases that stranglehold it has on ATF6 so it can go to the Golgi. I think it's actually an advantageous thing that it's got a relatively brief pharmacodynamic profile because it reduces the possibility of toxicity. And when we need it, we can administer it, and then it's kind of like on and then gone. So that brief window of time, we just need enough to induce the genetic program enough to provide the protection. Now, as you know, you just got to try it and figure out if it works. And then you figure out later, I guess it was enough. What was enough? So it turns out very subtle changes in the ATF6 transcriptome can cause these really great protective responses. As a matter of fact, if they were less than subtle and more high impact changes, it could be that it's not short-term and it's not actually always protective. So it's almost like a rheostat. Because it's so fleeting in its half-life, we can easily regulate how long the response is. So having said that, what we don't know is how fast it washes out of the ER. And its receptor is not ATF6, it's these protein disulfide isomerases.
Okay, thank you.
Okay, so I'm going to go with only one more question. We'll see here. Subnita Shadow has a question about if there are any physiological or pathological outcomes of the UPR that are not directly related to protein folding stress.
So was there any outcomes of the UPR? Yeah, so this is actually one of the most exciting things that our research has led to, and that is it is called the ER stress response, but it's more global than just the ER. And the ER is quite big in itself in many cells. So for example, the catalase, which I mentioned on my slide, but I didn't say, catalase is actually targeted to peroxisomes. That's where it generally lives, not in the ER. And so that's just one of many other gene products that we found that expand outside the ER. One of them I just want to mention is the Mtor complex, where we found that ATF6 regulates the expression of Reb, which is a protein required for Mtor activation. So when a cell goes to grow, it needs ATF6 there to facilitate enough Mtor activation for that growth process. This makes sense because when cells grow, you increase the amount of proteins, and therefore the protein folding requirements go up. So ATF6 is notified to turn on these things that help proteins fold, decrease reactive oxygen species, and many other things. So that's just two of these very fascinating things we found about the ER stress response in general, but ATF6 specifically.
Great. And there's one more question, final question here. Warren Weiner asked if compound 147 is being further developed as a drug, or if it's not, what are the limitations? If you can speak to that, I don't know.
147 probably won't be the compound that we take to much further development. It's a proof of principle compound. And we're in the process of doing additional screening, which Celina, you yourself know about, because we're collaborating with you and the center to do these screenings, where we might find new compounds that we can move forward with. That compound that I told you about today is a member of the Scripps small molecule library, and this limits what we can do with it in terms of further development beyond research setting only. But it's a proof of principle, and it could be altered or something else could be discovered that's a lot like it or works the same way. Now that we know how 147 works, we could maybe hone the screening process around that mechanism and get to new compounds more quickly.
Great. So we're going to end now because we are kind of over time now. I just want to thank you, Chris, again for a fantastic talk. Super exciting role of the UPR that is really not out there yet. So,
thanks, Celina. Thank you.
Yeah, so this is great. We're looking forward to working with you within ACDD. So now we have a 10 minutes break now. And I want to, before we go out, I really want to thank Dr. Schnellmann, our dean, and Dr. Sweezy, the head of the Cancer Center, for their support to ACDD and to this event. And also Liz Meller and Michelle Vogan for running this very smoothly. So stay tuned. We have another set of amazing talks coming up. We're gonna come back at, let's say here, 11:15. Is that correct? Yeah, so 5 minutes really. Thank you. See you soon.